Welcome everyone, and thank you for joining us. Our webinar today is entitled Understanding the Clinical Significance of the Commensal Bacteria. Our guest speaker is Dr. Christine Stubbe. My name is Dr. Patty Devers. I'm the Content Manager and a Medical Education Specialist in Genova's Asheville branch. I'm going to serve as the moderator for today's webinar. We would like to welcome Dr. Christine Stubbe. Dr. Stubbe received her doctorate of naturopathic medicine from Bastyr University in Kenmore, Washington. She completed a 3-year family practice oncology residency at Yellowstone Naturopathic Clinic and Frontier Cancer Center in Billings, Montana. Dr. Stubbe has also been trained by the Institute of Functional Medicine. In addition to participating in many teaching and speaking events, she has written for peer-reviewed journals and other publications. Dr. Stubbe currently works at Genova Diagnostics in our Medical Affairs Department as a medical education specialist and a content writer.
Thanks for showing up today, everybody. I'm excited to talk about the commensal bacteria. We get asked a lot of questions about our commensal bacteria, which is what prompted me to want to do this webinar today. So we have a lot to cover. Let's just dive right in. Our learning objectives are to get an overview of the 24 commensal bacteria that we measure on our GIFX profile and recognize some patterns that we see in those 24 commensals. We're going to meet a couple of key players, including Akkermansia, Methanobrevibacter, Smithii, and others. And then we're going to talk about how to balance everything out. So what is a commensal bacteria? This is the normal flora or normal microbiota that would be expected to be present in somebody's GI tract. So under normal circumstances, it would not be expected to cause any kind of disease. The microbiota are the actual microorganisms themselves in the defined environment. So today we're talking about the GI tract microbiota in a human. The microbiome is the entire habitat of microorganisms and their genes and the surrounding environmental conditions. So really the whole ecosystem. And then a dysbiosis would be an alteration in the composition of the commensal gut microbiota that can potentially harm the host. Over on the right, you'll see the 24 commensal bacteria that we measure on our GIFX comprehensive and GIFX microbial ecology profiles. And at the bottom of the page, we have the commensal balance and relative abundance charts that utilize algorithms to synthesize those 24 organisms that we see on the commensals page. This is really a small snapshot of a much larger picture, but it's enough to give us an idea of what's going on in somebody's microbiome. The GIFX doesn't represent all organisms that would be expected to be present, an entire microbiome, only the selected 24 targets, which covers a portion of the microbiome. In reality, the gut microbiota is represented by more than 1,000 microbial species. Why did we choose these 24? Well, we started with the literature and started looking in there to see what organisms have any sort of clinical associations, so disease states. And then we gave that list to the lab, and whatever they were able to validate is what we ultimately ended up with. And so we have genus and species level probes. And over to the right, I have a reminder here of the taxonomy. Maybe we remember this from middle school biology class, where we have kingdom, phylum, class, order, family, genus, species. So our probes, our DNA probes, are the genus and species level probes. And we based our reference ranges on a healthy questionnaire qualified cohort with other strict exclusion criteria applied to that. I have a couple examples here of taxonomy. So on the left, we see Canis lupus, the genus and species for dogs. There's actually close to 40 subspecies of Canis lupus, one of which is familiaris, which would be dogs, but the others are wolves and dingoes and other canine type animals. And we know that there are hundreds of dog breeds and they each have different personalities, like the Dachshund and St. Bernard. So just like there are different wolf subspecies and dog breeds, there are different strains of E. coli. So over here I have the taxonomic categorization of Escherichia coli, which most people are familiar with. And a lot of people are familiar with it or have heard of E. coli because of the toxic outbreaks with pathogenic Shiga toxin producing E. coli. But we do also have a lot of harmless commensal E. coli strains as well. The markers on our commensal bacteria page are grouped into 7 phyla. So the 4 major microbial phyla are the Bacteroides phylum, Firmicutes, Actinobacteria, and Proteobacteria, and that makes up about 98% of the intestinal microbiota. We also measure the Urarchaeota phylum, Fusobacteria, and Verrucomicrobia phylum. The organisms are grouped to phyla as a method of organizing the information taxonomically. They're not necessarily grouped according to function. Our brains really like to organize things discretely into groups so we can better understand them. And there's an attempt at this in the literature to try to find patterns. So they look at phyla, one phyla against another and create ratios, or they look at enterotypes. There have been 3 enterotypes that have been identified, which are just clusters of bacteria that trend together. But nothing is conclusive yet in the literature as far as these groupings. If you see the scientific name of the genus and species listed here, then we're only measuring that specific organism. So at the very top of the page, for example, we have Bacteroides vulgatus. So that's just looking at that one organism. If you see the genus followed by SPP, so I'm pointing to Odorobacter SPP here, then we're measuring, it's a genus level probe, and it picks up a broader set of organisms. It may not identify every single organism in that group. So some genuses of bacteria, such as Odorobacter, only contains a few species, whereas the Clostridium genus that I'm pointing to here has more than 100 species. So we don't have 100 Clostridium probes, we have 1 Clostridium species probe. And sometimes it's difficult for the lab to get these probes down to the individual species level. I'm going to switch gears here for a second. This slide is so important. It's probably my favorite slide of the whole presentation. It's going to be an anchor slide throughout the whole talk because we're talking about this concept of one big system. Not only is it important to look at the organisms themselves, but also to see what the organisms are producing. The microbiome has actually been described as another organ in the body that has all types of functions, and the microbiota themselves actually weigh about 2 to 6 pounds. What this picture is showing, if we look in the middle here, we have the ecosystem, which is a picture of our intestines. And the ecosystem is influenced at the top left by the diet, so what we're eating and putting into our gut. And at the bottom, there's host mediators. So the brain produces neurotransmitters. The liver is contributing bile salts. The intestinal barrier has mucin products and other things that are creating an ecosystem. And the immune system interfaces as well. So this ecosystem shapes and composes the bacteria that live in our gut. And those bacteria are in turn going to express certain genes that produce metabolites. And over on the right, we have in the blue box a list of just a few metabolites that are produced. There's actually close to 1,000 metabolites that have been discovered and counting. And they have varying effects ranging from beneficial, like the short chain fatty acids, to problematic, like TMAO, that leads to atherosclerosis. These metabolites impact the human. So up at the top, you can see that it sends out signaling molecules to the human body, and it impacts our metabolism. It can impact inflammation pathways, so they're very, very important. These bacterial metabolites can be detected in the stool, in the blood, urine, even in our breath. And we do have some other tests that detect some of these other metabolites other than stool, but we're not going to get into that today. Another important part of this diagram is what I just outlined here in the red box. These metabolites in turn go back and influence the gut, which is the ecosystem that these bacteria live in, and that in turn is going to influence genetic expression in the metabolites that get produced. Now, if we think about bacteria and genetic expression, all genes are not turned on all the time and getting expressed all the time. The environment is going to turn on or off certain genes and tell it what to express. Same thing with the human. All of the genes in our entire genome are not being expressed 24/7. My microbiome made me do it. So who's really in control here? Is it us or our microbiome? In reality, it's a two-way street. What you feed yourself ultimately feeds your bacteria, and then they produce metabolites that affect us. The bacteria in our gut actually outnumber the number of cells we have in our body, so we better treat them nicely. The gut-brain connection is huge in the literature right now, and it can affect our mood, behavior, cognition, and there's actually a class of probiotics that are created for people with mood disorders, and they're termed psychobiotics. Short-chain fatty acids are postbiotic metabolites. So this is a term that I'm seeing more and more now is postbiotic. So a lot of people have heard about prebiotics, which are fibers that are supporting the beneficial microbes, and then probiotics are the beneficial microbes themselves, and they ferment the fiber and produce these short-chain fatty acids, which are postbiotic metabolites. We measure short-chain fatty acids on our GIFX test. We look at butyrate, acetate, and propionate, 3 short-chain fatty acids. Butyrate's really important because it feeds the colon epithelial cells, and acetate and propionate feed other bacteria. They're involved in lipogenesis and gluconeogenesis. And ultimately, the production of short-chain fatty acids lowers the pH in the intestines, and that's what keeps the pathogens and pathobionts at bay. So we don't just look at the bugs on this test, but we also look at what they produce to try to see the bigger imbalance picture. And in addition to short-chain fatty acids, we also look at products of protein breakdown, beta-glucuronidase. We look at secondary bile acids on our CDSA 2.0 test. I really like this slide because it shows us how the bacteria depend on each other to function. This example shown here is referred to as cross-feeding. And so you can see that there's substrate that's getting degraded by these primary degraders, Bifidobacterium and Ruminococcus, that we measure on our 24 Commensal page. And then they get converted to intermediate products, and the butyrate producers then feed off of that to produce butyrate. So you have Faecalibacterium and Rosaburia, also ones that we measure on our 24 Commensal page. So if you didn't have these primary degraders, you might have the butyrate producers, but they might not be able to produce butyrate because they're missing their buddies that help them out. What story is our microbiome telling us? So the Human Microbiome Project started in 2008. So this is just over 10 years ago, which in literature and science research terms, that's not a long time. They set out to map out the entire human microbiome. So it takes time to study and make observations and conclusions. So that's a work in progress. It's kind of in its infancy right now. Genova has developed a system of data analysis based on a very large database of close to 200,000 stool tests. So we're going to look at some of that data analysis now. But we don't want to get caught in the details of individual organisms or metabolites and not be able to see the bigger picture, which is similar to this picture that we see here, not being able to see the entire forest when you're focused on one tree. We can't really look at the Commensal page or one marker in isolation and expect to solve the problem. The microbiome is a whole lot more complex than that. It's important to recognize certain patterns to translate the meaning, and we need a computerized algorithm to do this computation for us. Unless you have a brain that works in algorithms, it's not really possible to just make sense of the different levels of organisms. It needs to be synthesized. So we perform a data analysis on our unique set of markers versus extrapolating from the literature, because in the literature, all studies don't necessarily look at the same set of microbes, so it's not really an apples to apples comparison. They might use different methodologies, and there's not really a standardized way to assess the microbiome at this point. And with all those things, you might see different conclusions. So the literature provides a starting point. From there, we take more information, like information from patient questionnaires. We do an analysis of biomarker associations with each other, even across different tests. And the goal is to create meaningful data unique to our set of microbes. The commensal balance chart. We have other webinars and support guides that go into full detail about the clinical associations charts, the commensal balance chart, the relative abundance. So I encourage you guys to look into that one for more information. But just really quick, Genova likes to have colorimetric reporting, meaning green, yellow, red. Green and yellow is generally where you want to get your patient, with green being the optimal zone and red being outside of range. And so here we're aiming for green, so that would mean better commensal balance. We also look at a relative abundance graphic, so that's at the top of the page here, and you can see in this example the patient results are quite a bit lower as compared to the healthy cohort. And what we found is that lower commensal abundance is associated with inflammatory biomarkers and a higher inflammation-associated dysbiosis score. In our data analysis, we identified two different clusters, a high commensal balance cluster, which might be associated with bacterial overgrowth. We're not going to talk about that one today. And cluster 2, which is the low commensal balance cluster, that's associated with these inflammatory markers, calprotectin, EPX, and secretory IgA. We just published this study, the references at the bottom of the page. Over on the right, I really like this chart because it illustrates nicely that as the inflammation-associated dysbiosis score groups increased from left to right, so we're at the x-axis here at the bottom of the screen, so lower inflammation to higher inflammation, you could see that the fecal EPX, which is eosinophil protein X, calprotectin here, and secretory IgA all increased as the inflammation-associated dysbiosis score increased. The inflammation-associated dysbiosis score is an algorithm composed of the individual commensal bacteria themselves and one other biomarker. And so it's truly a bacterial dysbiosis score. And at the top of the page, you can see that when the IAD score is low, meaning less inflammation, the commensal abundance is higher or normal, and the abundance drops as inflammation goes up. These are our 24 commensal bacteria, and all of these organisms together comprise the inflammation-associated dysbiosis score, not just one in isolation. The IAD score was verified in 2 clinical studies with patients who had inflammatory bowel disease as compared to non-inflammatory conditions like IBS or celiac or healthy patients. In the UCLA study on IBD patients, the algorithm-based score was able to show statistical significance while all individual microbes failed. That's a really important point because it's not like you can look at one of these bugs and say, oh, it's inflammatory, this is bad. It's more about the whole community working together. And that's what this paper, one of their main points was that the pathogenic community, no single microbe is pathogenic alone. So I'll read some of it here. It says, in addition to the classic pathogenic species, we propose that another kind of pathogenicity exists in the gut, one in which the whole community is pathogenic when its emergent properties contribute to disease. In a pathogenic community, no single microbe is pathogenic alone. Instead, the community assemblage is an environmental risk factor that contributes to a disease state. So it's really the whole environment that makes the community pathogenic. This is another analysis that we performed based on patient questionnaires, and you can see how our healthy cohort differs from the disease cohorts. There's 8 disease cohorts listed here. The difference is with the blue arrows, so they are either statistically significantly higher or lower in that particular commensal bacteria as compared to the healthy cohort. So this page isn't necessarily meant to diagnose these conditions or predict risk for these conditions. It's just showing that there's an association with essentially a dysbiosis, and conditions like IBS and IBD, gut conditions, as well as outside of the gut conditions like chronic fatigue, mood disorders, high blood pressure. And there's literature associations beyond these 8 that we looked at, like autism, eczema, Parkinson's, multiple sclerosis. So the gut really plays a major role in most disease processes. So now we are going to jump into those commensal bacteria, the individual commensals. We don't necessarily have time to go through all 24 today, but we will highlight the main ones that we get asked questions about. We are working really hard on a document to outline each of these 24, so you'll have that to refer to in the future. Today's talk is really about literature-based information and human studies, and in some cases, animal intervention studies and human intervention studies conflicted, so that's why I didn't include animal information. And just a side note, in reading and preparing for this webinar today, I made an observation. There is so much conflicting information, even with the human studies, and it's challenging to find consistency among the studies. So I'm just presenting the information that I was able to find. So starting at the top of the page, we have the Bacteroides species. I want to make a note here of the difference in just 2 letters. So we talked about the Bacteroidetes phylum, and we have Bacteroides species. So it's a difference of 2 letters, a T and an E. And the reason I mention that is because in preparing for this talk, they were used interchangeably sometimes, or maybe it was a typo. And so that's something to pay attention to, because a phylum level assessment is way different than a genus level assessment. So the Bacteroidetes, so I'm talking about phylum level here, is a very versatile group. It's composed of different players that do sometimes completely different things from each other. The Bacteroides genus that we're talking about is capable of feeding off of lots of different substrates. So it says here the Bacteroides enterotype correlated with diets rich in animal protein and fat. So we can't always make generalizations and jump to conclusions, because the Bacteroides species, different species, also feed off of different substrates, even plants. So they can eat complex carbohydrates and simple carbohydrates too. Some Bacteroides contain lipopolysaccharide, which is an endotoxin that's produced. They also produce beta-glucuronidase, secondary bile acids, short-chain fatty acids, products of protein, breakdown, and vitamins. So this group does a lot. They're associated with reduced microbial diversity in one paper. Insulin resistance, that's specifically referring to Bacteroides vulgatus, which is the second PCR target that we measure on this page. It's been shown to be increased with red wine, animal-based diets, high-fat diets. Just a comment on fat in studies. A lot of studies don't really talk about the quality of fats, and I wish that they would do that more. Mainly, I think a lot of the time they mean fat from a Western diet, like fried foods, animal fat, trans fats, so the bad fats, but they don't always really specify. We know that fat influences our inflammation. We have the anti-inflammatory omega-3s and pro-inflammatory arachidonic acid from animal fat. So I think it's important to specify, even though they don't always specify. And then Bacteroides were shown to be decreased with tart cherry juice, inulin, green tea, and dates. The next target that we're looking at here is Prevotella species. The literature's mixed on the association with inflammation. Some says pro-inflammatory, others says anti-inflammatory. The Prevotella group produces short-chain fatty acids, acetate and propionate. The Prevotella enterotype is richer in response to plant-based diets and complex carbohydrates. Now, the enterotypes, as I mentioned, there's a landmark paper published in Nature on the 3 enterotypes. And one of them was Bacteroides that we just looked at. The other was Prevotella. And then the third one was Ruminococcus, which I'm not really going to talk about today. And so these are generalizations that they saw in these 3 enterotypes, that Bacteroides was more animal-based, Prevotella more plant-based. And they saw Bacteroides more in the Western population, whereas they saw the plant-based more in an African population that was eating more plant-based foods with the Prevotella enterotype. And so the enterotypes and our bacterial composition can vary based on where we are in the world, based on our diet. And that group that published the enterotypes paper had a follow-up paper where they said it's still too early to draw broad conclusions. So Prevotella is increased with a plant-based diet, decreased on a Western diet. Western diet in general is defined as high fat, high sugar, low fiber, also referred to as the standard American diet or the SAD diet, unfortunately. Next on the list, we have Clostridium species. This genus contains more than 100 species. And I don't want you to get confused with the pathogenic Clostridium, that things like Clostridium difficile. We have an add-on test for C. diff, so this probe is not meant to diagnose pathogenic Clostridium infections. Clostridium produces short-chain fatty acids, hydrogen, beta-glucuronidase, and secondary bile acids, and it's necessary for immune homeostasis. Notice the numbers off to the right with the reference ranges. Clostridium is the highest compared to all these, with the e to the 10th power here. So you can see some of the other ones are e to the 6, e to the 7, e to the 9th. So this was the highest with e to the 10th. It's increased with coffee and high beef diets, decreased with Mediterranean diets and different prebiotics. Faecalibacterium prausnitzii is one of my favorites. It's decreased in intestinal disorders like Crohn's disease, ulcerative colitis, colorectal cancer. So we want to have a good amount of it. It's anti-inflammatory and supports healthy intestinal barrier function. Genova actually found a negative association with all 3 inflammatory markers and Faecalibacterium. So remember, in this case, a negative association is a good thing. The higher the Faecalibacterium, the lower the inflammatory markers. It's the body's main butyrate producer, and it produces B vitamins. It's increased with inulin-type fructan and FOS, prebiotics, fiber, apple pectin. Look at that, physical activity. So moving beyond diet, there's other things that can be associated with an increased level. It's decreased with gluten-free diets and a low FODMAPs diet or excess bile salts. The FODMAPs diet is prescribed a lot for functional gastrointestinal disorders like IBS or SIBO, small intestine bacterial overgrowth. Those diets are not meant to be long-term. Lactobacillus and Bifidobacterium. So this is a very busy slide here. I realize I'm not going to get into depth about these beneficial bacteria. They're seen in a lot of probiotic formulations and fermented foods. Their beneficial effects are widespread, and we could actually do a whole course on them. Most people are aware of them because they've seen them before and they're on their yogurt label. There's over 50 species of Bifidobacterium and 100 species of Lactobacillus, and they can do things like produce antimicrobial peptides against pathogens and potential pathogens. They can regulate virulence gene expression. Bifidobacterium longum, which is one of the bacteria that we species that we actually measure here, enhances secretory IgA in the gut and their response to antigens, so that's a good thing. Dr. Olmsted actually did a whole webinar in our webinar archive on probiotics, so I encourage you guys to listen to that webinar as well. The Prateabacteria phylum is in general considered a pro-inflammatory phylum. So when it's in normal proportions, it's fine. This phyla contains a lot of the gram-negative pathogenic species and some producers of LPS. So Klebsiella, Citrobacter, Proteus, these are things that we culture and we see a lot of the time. Shigella that we culture, or H. pylori that we run as an add-on test, they all belong to the Prateabacteria phylum. So over in this picture, over on the left at the bottom, we see the blue healthy as it moves into the red disease state over on the right. And right above that, this little triangle here is showing high community stability, so that's the bacterial community, and low community stability on the right. So we have a healthy person who has very little proteobacteria. As we move to the right, neonates, we're not going to really talk about infant microbiomes today because they're completely different than an adult microbiome. Today we're really focusing more on adults. So, you know, you can see that the neonate has proteobacteria, but they're still high community stability and they're healthy. Gastric bypass, so that can alter the pH, bile flow, and intestinal hormones that influence the abundance of proteobacteria. And then we see metabolic disorders, inflammation, and cancer have higher proportions of proteobacteria. The paper that's cited there quotes a few mouse studies saying that artificial sweeteners and food emulsifiers induced a proteobacteria bloom, and that low fiber diets and inflammation are hypothesized to provoke the growth of this phylum. But we don't want to make generalizations that they're all bad. Let's look at some of the organisms that we measure here. We look at Disulfovibrio piger, and it's a hydrogen sulfide producer. It can actually decrease butyrate. Some other sulfate-reducing bacteria that we measure on our test include Fusobacterium, which is a little farther down on the report. It's associated with inflammation. Disulfovibrio is increased with IBD, colorectal cancer, and visceral nerve sensitivity. In animal studies, it was increased with a high-fat, high-sugar diet and chondroitin sulfate, which is a supplement. I couldn't find any human studies as to what decreases or increases disulfovibrio. It's resistant to most antibiotics, so while the antibiotics are out killing the pathogen and the good guys, D. piger stays and can become a pathobiont. So this is not a good thing. Disulfovibrio is decreased with Lactobacillus plantarum. The study with Lactobacillus plantarum was on humans where they took the probiotic for 4 weeks and saw a statistically significant decrease in the D. piger bacteria. So I want to touch on this concept for a minute. A lot of people think they have to go in and kill, and I just think of the mass destruction that the killing causes, because when we have antibiotics or antimicrobial herbs, they're not targeted to only address this one organism. They're going to kill other things as well. It's really destructive to the rest of the microbiome. And what I think is going to happen over time is that treatment might shift more towards creating harmony by adding in more of the good guys to have a positive influence on the unruly ones, and more importantly, changing the overall environment to not be so hostile to our fragile microbiome. We'll talk about that a little bit more in a minute. Also, in vitro studies showed that a lower pH was associated with decreased levels of D. pigar. And so remember, a lower pH can be created by increasing those beneficial short-chain fatty acids. And some clinicians have mentioned using bismuth or zinc acetate to address this. Those things can bind to hydrogen sulfide gas, but I couldn't find any human studies on this. All right, E. coli. This group has many strains. Most do not cause disease. The disease-causing strain that people have heard of, the Shiga E. coli, is something that we have as an add-on to this test. It synthesizes vitamins, ferments amino acids. It can produce ethanol and promote gut permeability. It's increased with gluten-free diets. It consumes simple sugars, decreased with a plant-based diet and probiotic yogurt. And then there's this E. coli Niceal probiotic. This probiotic was studied with preventing disease relapse and ulcerative colitis. It protects against pathogens and maintains tight junctions. So again, not all E. coli is bad. We're still in the Prateabacteria phylum here, and we have this bacteria called Oxalobacter formigenes. And this is the key bacterium responsible for the degradation of oxalate. So it reduces oxalate absorption into the gut and excretion into the urine, so ultimately reducing the risk of kidney stones. Oxalate is in a few different foods like spinach, rhubarb, almonds, cashews. The list is there. We don't know a lot about when or how people become colonized with Oxalobacter, or even the persistence of it over time. It's present in very low relative abundance to everything else, and it's not really a core component of microbial communities despite its relevant function. It's very sensitive to and reduced with commonly used antibiotics. And Lactobacillus and Bifidobacterium are also capable of consuming oxalate. So the picture at the bottom shows where oxalate and calcium come from in our diet, ultimately to form those calcium oxalate stones. And this picture in the middle touches on the fact that the microbiota don't really act in isolation. It's a community. And so Oxalobacter and other oxalate degrading bacteria all work together to reduce that oxalate absorption to the kidney and ultimately decrease the risk of kidney stones. The next organism is Methanobrevibacter smithii. It's a hydrogen-consuming, methane-producing archaea. So an archaea is not really even a bacteria. I know it's on the Commensal bacteria page, but we weren't going to name it Commensal bacteria plus Methanobrevibacter. Methane gas is actually associated with obesity, prediabetes, and constipation. Methane gas itself may slow the intestinal transit, leading to constipation. And it's shown to be decreased with rifaximin plus neomycin. This was a smaller study on 11 pre-diabetic obese patients who were treated with these two antibiotics. And not only did they eradicate the Methanobrevibacter smithii and also lower breath methane levels, but they also improved LDL, total cholesterol, and insulin levels. Some other things that have been shown to decrease it are statins, probiotics with Lactobacillus and Bifidobacterium strains. And then one study showed garlic oil decreased breath methane levels, but they didn't mention this organism. Now, the majority of methane in the human gut will be produced by this Methanobrevibacter smithii. This is a picture of our breath test that we offer here, where we measure both hydrogen and methane gas. And we have seen a correlation with Methanobrevibacter smithii and breath methane levels in patients who ran both the stool test and the SIBO breath test. And the last organism I'm going to talk about is probably my favorite. It's Akkermansia muciniphila. Low levels are associated with a bunch of metabolic conditions. So let's look at the picture over on the right. On the left of the picture, you have a picture of the gut. And in patients with obesity, alcohol consumption, intestinal inflammation, you can see that the mucus thickness is thinner, and the junctions, the tight junctions between the intestinal cells are a little bit permeable. So things like LPS can get through and lead to metabolic endotoxemia, and it's pointing to some disease conditions here: hepatic steatosis or fatty liver, and inflammation, insulin resistance, type 2 diabetes. Now, over on the right, if you have an abundant amount of Akkermansia muciniphila, which is associated with a thicker mucus level that protects those intestinal cells, then you're not going to have that permeability and less likely to see these conditions. So Akkermansia is a mucin degrader. It produces acetate and propionate, which are short-chain fatty acids. It improves intestinal barrier integrity. It may limit toxicity of sulfate-reducing bacteria like that Desulfovibrio piger that we looked at. And it's actually being studied as a probiotic for metabolic conditions. And Genova also saw that compared to the healthy cohort, all 8 of those conditions that we looked at earlier were associated with lower Akkermansia. So this is an important one to have. It's increased with pomegranate, caloric restriction, resveratrol. Again, here we're seeing the physical activity. Decreased with a low FODMAPs diet. So let's talk about strategies to balance the microbiome. We don't necessarily have a supplement for each bacteria that is measured besides Lactobacillus and Bifidobacterium. The Commensal page was not meant to result in a customized formulation of probiotic bacteria to give to the patient. And it's difficult to make specific recommendations because of the complexity of the microbiome. So the suggestions are going to be based on literature and general treatment strategies that are thought to create overall balance. We also get feedback from our practitioners on what they like to use to see improvement clinically and on their test results. So today I also want to focus on things that are modifiable. There are things that impact the microbiome like birth method, whether it was vaginal delivery or C-section, or breastfed versus bottle fed. And most of us are past that now, so we have to focus on how to keep it healthy now. So this slide, this paper's conclusion is that the Western diet, which is the low fiber, high fat, high carb diet, is a factor that can lead to severe dysbiosis. In contrast, the Mediterranean and vegetarian diets that include abundant fruits, vegetables, olive oil, and oily fish, all anti-inflammatory things, are known for their anti-inflammatory effects and could prevent dysbiosis and subsequent inflammatory bowel disease. A plant-based diet appears to be beneficial for human health by promoting the development of a more diverse gut microbial system, that's important, or even distribution of different species. So does this mean everyone has to become a vegetarian? No, we all just need to eat more plants. And the diversity concept is promoted with variety. So a variety of foods is going to promote the growth of the different organisms and thus the production of the different metabolites that influence the microbiome environment. We already saw that physical activity can help. It helps by increasing gut microbial diversity. And these particular species, Faecalibacterium, Rosaburia, and Akkermansia. And I just saw a study on whole body vibration, and this was done in mice, and that showed a beneficial effect on the microbiome and inflammation. So who knew? And then stress, we know, is not good for anything. Stress actually decreases beneficial lactobacillus and bifidobacterium, and it has a negative impact on all these different functions of the gut here in this picture at the bottom of the slide. Medications can impact the microbiome balance, so antibiotics, PPIs, NSAIDs, and others. Antibiotics are probably the top disruptor of the microbiome, and I just think of antibiotic stewardship. We want to make sure that antibiotics are being taken for the right reasons, and I feel like there's a lot of unnecessary prescribing out there. I mean, who in the audience hasn't been on an antibiotic? I think most people have. And so this is widespread and potentially why we have a lot of health conditions nowadays. It actually can take up to 2 years to restore the microbiome with some antibiotics. Even antimicrobial herbs can wipe out a large portion of the microbiome. PPIs are associated with decreased microbiome diversity and an increased risk of SIBO. And then we have a list of other toxic exposures here as well. So I'm not going to go through each one of these today, but sugar, pesticides, chlorinated drinking water, artificial sweeteners. I forgot to add smoking to this. There's a study showing it's associated with decreased diversity. So this is a list of a few fermented foods. So if you want to work on your microbiome, you can get it from food, you can get it from supplements. So here I'm talking about the food. These different foods like fermented dairy, kombucha, miso, tempeh, they contain probiotics like lactobacillus and bifidobacterium and others. I don't want to get lost on this slide. The food for the microbiota, and which is mainly fibers and plants. People ask, you know, what type of fiber should I give? And the answer is a variety. Most foods are going to be a mix of soluble and insoluble fibers anyway. And rather than worry about the composition of the fibers, the take home here is that there are a lot of plants mentioned on this page. Just eat more plants. At the top, we have resistant starches, which come from things like unripe bananas, seeds, whole grains, and the fermentation of resistant starches is what produces those beneficial short-chain fatty acids. I have some food examples of inulin and FOS. Those are things we see on as prebiotics on supplement labels sometimes, and people might wonder, how can I get that in my diet? And then with soluble fibers, you know, we have pectin from apples, beta-glucan from oats. Beans are important. I want to mention beans for a second. If you guys are familiar with the blue zones around the world, these are the zones where folks live into their hundreds and they're healthy. They're not necessarily on medications. They don't have disease. And, you know, they've researched why. So this is going to be places like Okinawa, Japan, Sardinia, Italy, Loma Linda, California, Costa Rica. There's other places. And one commonality among all those places is that they consumed beans. So I know a lot of people avoid them because they produce gas, but if you have a healthy microbiome and give it a little time to balance out, then you might be able to tolerate them better over time. I also have on here plant polyphenols, and then just a point that special diets may lack prebiotics. And we see this a lot where the beneficial bacteria might look low on the report, or the short chain fatty acids are super low. Sometimes a paleo, low carb, ketogenic, grain-free, or low FODMAPs diet, which for the most part lack a lot of prebiotics, might produce a dysbiotic pattern. This page is just talking about supplements, so a list of things that you might see as prebiotics and supplements are at the top. Probiotics would include lactobacillus, bifidobacterium, Saccharomyces, spore-based, and others. There's really no consensus on probiotics and dosing. Some clinicians rotate probiotics. There are online databases that show conditions and strains studied, things like Probiotic Advisor or usprobioticguide.com are some examples. And then there's postbiotic supplements out there. So butyrate, which is a short chain fatty acid, has been studied with inflammatory bowel disease. Now, butyrate naturally occurs in butter. Remember, the bacteria manufacture butyrate with the right substrate, so the prebiotic fibers. So you can count on your own microbiome to produce it, or you can take it as a supplement too. These other parts of the test are part of the microbiome too. So we have 4 EIA add-on tests for pathogenic bacteria on the left here. And then we culture organisms and identify them through our MALDI-TOV machine library. And the question again, do we always have to kill everything? Well, the true pathogens probably, but anything that's potentially pathogenic, it's always a question. If we kill them, are we promoting that dysbiosis pattern even more by giving antibiotics? So that's always a fine balance. Use your clinical judgment there. But some probiotics produce antimicrobial peptides. Some clinicians use things like colostrum, which is rich in IgG and promotes secretory IgA, which helps protect against infection. IgA actually binds the bacteria. So think about immune support and our natural defenses too. Saccharomyces boulardii is a beneficial yeast probiotic that bolsters secretory IgA levels. So rather than having this killing focused mindset all the time, think about ways to improve the environment. And we're going to look at other test findings. This is part of the environment that these organisms live in, address the inflammation and the undigested food particles. This is the larger ecosystem. Some bacteria thrive in a high-fat, inflamed environment and others don't. How would you like it if you were in an inflamed environment? You might act a little differently. You might start expressing different genes. So consider that holistic approach. Repair the gut, repopulate with a 5R approach, remove irritants, replace or replete, rebalance the lifestyle. Some final key points here. Organisms don't act in isolation. It's not a one bug, one treatment approach. If you recommend an intervention for one bug, it probably won't just influence that one bug. We don't really know the greater impact it's going to have on each of the other organisms. Is it good? Is it bad? We don't necessarily know that. Therapeutic interventions like dietary macronutrient content, fiber, prebiotics, probiotics, lifestyle modification, and the environment, so everything, has been shown to modulate the individual microbiome. And just remember that microbial diversity is a good thing. So food diversity can promote microbial diversity. Diet continues to be the most important determinant in shaping the composition, diversity, and richness throughout adulthood. In general, intake of fruits, vegetables, fibers is associated with a higher richness and diversity of the gut microbiota. And then this other paper quoted A majority of the studies noted that protein consumption positively correlates with overall microbial diversity. They went on to say in that paper that the protein diversity relates both to plant and animal protein sources. Our microbiome does a lot to keep us healthy, and it's an extension of us, so we want to take care of it by taking good care of ourselves. All right, so that was a lot. I bet there's some questions out there. Dr. Devers, I'm ready to take any questions you might have gathered.
Wow, you're right. Thank you, Dr. Stubbe. That was a great presentation. So many questions came through. Let's just start with this one. And it's an important one that we get a lot in medical affairs. Can you speak to the clinical significance of the Firmicutes Bacteroides ratio, which is found at the bottom of that commensal page?
Sure. So originally, when we came out with this test, we included that because the literature suggested that a high Firmicutes Bacteroides ratio may be associated with a greater risk of metabolic syndrome, diabetes, obesity. So that's why we introduced it initially. However, like a lot of studies, the literature is mixed in this subject. So some papers say a high FB ratio, some say low. Not all sources using the same methodology or even the same set of markers. So we're going to continue to do an analysis of our markers. But in the meantime, the Firmicutes and Bacteroides phyla are major phyla that make up the microbiome. And so when I see them out of balance, either high or low, that just speaks to an overall imbalance picture. And we want to use those strategies for balancing that I talked about.
Great, great, thank you. Could you also speak to the difference between the levels of lactobacillus and E. coli on the PCR page versus the discrepancies that we often see in culture?
Sure, yeah, that's another common question that we get. So we're looking at two different methodologies here. So anytime you have two methodologies, it's bound to have a little bit of discrepancy. It's with lactobacillus in particular, that's the one that I see below detectable limits, or it won't grow out on the culture, but you might see it on the PCR. It's hard to culture lactobacillus. It's a facultative anaerobe, and what that means is that an anaerobe thrives in an oxygen poor environment, but facultative means it can go either way, but technically it's an anaerobe at heart. So when you're growing lactobacillus in aerobic culture conditions, sometimes it's hard to grow out. Remember, there's hundreds of lactobacillus species, and they all act differently. So some might culture well, others might not. Ultimately, it's good to have more than one methodology in case one picks it up and the other doesn't, which is why we do it that way. We have both the PCR and the culture.
Great, thank you. In the interest of time, we're going to end our question and answer period there. For additional educational materials, we'd like to encourage you to visit our website, gdx.net, and/or contact client services. We also offer complimentary appointments with our medical education specialists to answer questions related to our testing, including choosing the right test and reviewing patient test results. Finally, we'd like to encourage you to register for our upcoming webinars on our website. Next month, we will be rebroadcasting a webinar presented by Dr. Deanna Minnick entitled Fatty Acids, Making Clinical Sense of Lab Reports. Thanks again to Dr. Stubbe, and happy birthday. Have a great day, everybody.
Thank you so much.